thank you everyone for joining us for Dauphin Island Sea Lab's research seminar series this Friday. And I hope everyone is recovering well from power outages and everything else over the last 24 hours. But we are very fortunate today to have Dr. Christina Toms with us. She's a research associate with the Chicago Zoological Society's Sarasota Dolphin Research Program. And she has been a colleague who's worked with us here on the Northern Gulf Coast for many years. I've actually known Christina since she was doing her master's degree at USM. But she got her bachelor's as a double major in marine science and psychology from the University of Hawaii, which sounds so cool. And then her MA from USM in 2011 working with dolphins. And then her PhD in conservation biology, also working with dolphins at the University of Central Florida. So thank you so much for joining us and telling us a little bit about your ongoing work.
Thanks for having me, and thank you all for those of you who are here listening. I appreciate it. I hope all of your families are safe after your hurricane recently, well, the many hurricanes recently, especially the one yesterday. So thank you for being here. Again, my name is Christina, and I'm going to be talking today mostly about my dissertation research. I just graduated last year, and this is the first time I've actually been able to talk about it since my defense, so that's kind of cool. I also haven't done one of these virtually, so I talk with my hands a lot, hopefully. We'll see how that goes. If you see my shoulders shaking while I'm talking, it's probably because my hands are moving and you can't see it. All right, so to start off with, I'd like to acknowledge all of my collaborators and funders and appreciate them for all of their support to get me through this. And I'm going to start with a little bit of background in general about the Marine Mammal Protection Act, because some of the policies and management legislation is kind of an important impetus to my project. And I'm not going to assume that anybody here or everybody here knows anything about marine mammals. So this was a really important piece of legislation that was implemented in 1972 and was the first to mandate an ecosystem-based approach to marine resource management, which was really important at the time. Ecosystem-based approaches to conservation management now is a lot more common in how we kind of tend to think about things, but at the time was very new, especially to be implemented. So an important part of this is that management units were defined as stocks, meaning that we may or may not have biological information about them, but we needed to be able to implement something to define units of measurement. And an important part of the language was that marine mammals should continue to be a functioning element of their ecosystem. That was kind of the big piece of the legislation that was an attempt to use marine mammals as an umbrella to manage an entire ecosystem instead of one species at a time or one taxonomic group at a time. And so that's kind of that's an important part as to why we do things the way we do things today. The important pieces of information for that is to define population units and then to assess the abundance for each of those units so that management can decide whether or not there are enough deaths or removals from a community to deem important for intervention. The biggest problem with any management system is data deficiency and having the numbers that are needed for management in order to actually make management decisions. Bottlenose dolphins in general are globally distributed throughout temperate and tropical waters and currently are recognized as 3 extant species. The ones that you're going to be most familiar with here in the US and the Gulf of Mexico are Tursiops truncatus, which is what we're going to focus on today. Throughout their range, there are 2 general ecotypes, as we call them. There's an offshore form and a coastal form. Originally, these were delineated based on diet, morphology, parasite loads, hematology. More recently through genetics, so there's genetic distinction between these 2 forms. I'm going to focus primarily on bay sounds and estuaries, and so the coastal form today, and that was the bulk of my research. They're very interesting in the fact that these bay sounds and estuaries tend to be home to year-round small resident communities of dolphins. Though they have the capacity to travel all over the place if they want to, in these small bay sounds and estuaries, they tend not to. There tends to be a resident community that lives in our backyard, so to speak. They have varying degrees of overlap socially with non-residents. So you might experience animals come into a region that you'll never see again, and we call those transients. We might have seasonal animals that come in and utilize a given geographic range for a little bit and leave again. Socially, they're really complex. We refer to their societies as fission fusion, which is basically a fancy term for saying They don't do anything consistently. They can hang out in family groups, but often you'll see moms with calves of the same age hanging out together. They come in together and break apart over days, hours, sometimes can be consistent. But generally, they don't have consistent social structures, except for males. In Tursiops truncatus species, male pairs tend to bond for life unless one dies, which is very consistent. The inshore groups are more susceptible to anthropogenic stressors, just because they live in our backyards where our presence is more influential. So they're subject to boat traffic and pollution runoff and flooding events and all the things that come with being close to shore. From a management perspective, there are 31 designated stocks in the Gulf of Mexico throughout the bay, sounds, and estuaries. So these are just the small inshore systems. These were originally delineated basically based on geographic units, not based on data. I mentioned that there's a lot of data deficiencies. And originally there were some aerial surveys to try and understand the abundance across these different regions. We now know that aerial surveys aren't particularly effective in capturing abundance at this scale. And at the start of my project, the only geographic areas that had data were these in yellow here on the map. And another important piece for management is that they're updated every 8 years in order for all of their policies to come together, if you will. And the only population genetics that had really been done at the beginning of my research was down in Sarasota, which is where I am now with the Sarasota Dolphin Research Program. Anna Celis had done her graduate work down here looking at fine scale structure. So despite the fact that they have capacity to move all over the place, she showed, and one of the first to show, that genetic structure can occur on very, very fine spatial scales. And at the time, the offshore kind of collective group in the Gulf of Mexico, the management units looked like this. It was based on bathymetric depths, assuming that dolphins would organize themselves sort of related to bathymetry. But we didn't have a lot of data to support that either. That was just based on assumptions to begin with. So then comes the Deepwater Horizon Oil Spill, which I'm sure many of you are familiar with, in 2010, which hit Louisiana, Mississippi, Alabama, and Florida very hard, especially among many organisms, but bottlenose dolphins were also very strongly impacted. Over 1,200 dolphins stranded, or cetaceans stranded, most of which were bottlenose dolphins, and most of which were the coastal form that I mentioned, which are the nearshore, inshore groups. The worst area for oiling of the spill, as you're probably familiar with, was from Louisiana through sort of Pensacola Bay, but that was the most eastern extent, not nearly as hard hit as the other areas. And our colleagues in NOAA started some projects over in Barataria Bay and Mississippi Sound. We didn't really have any baseline data to be able to evaluate the impact of such a major event like this on bottlenose dolphins. And the only ones in this region that really existed in terms of data before the event are the ones in yellow on the east coast here, or the eastern side of the Panhandle, sorry, and which wasn't particularly helpful with respect to the oil spill. And as we moved along, Ruth Carmichael's group here at Dolphin Island Sea Lab, we're starting to work in, well, they already had their stranding network going in Mobile Bay, but population dynamics was a work in progress. And my colleague, Steve Shippey, was doing his PhD at UCF in Choctawhatchee Bay, just outside the zone of the oiling. And so we needed to fill gaps in this area, and hence my research got started in Pensacola Bay. My colleague Steve had done a little bit of work in the southern reaches of the system, but we really didn't know much about the population beyond that. So my dissertation was built on building capacity, basically trying to build data gaps within the northern Gulf of Mexico so that we could start answering these questions in the future about impacts such as an oil spill or hurricanes or flood events, because you need data, baseline data, to be able to do that. My approach was focused on sort of two major scales, both an ecological scale, looking at population dynamics, and then looking at an evolutionary scale at fine scale population genetic structure across the Florida Panhandle. So the rest of my talk is going to be split up into those first two, population dynamics first. We're going to talk about genetic population structure second. And then I'm going to talk to you a little bit about a case study that came out of my research as well, which was a side project that turned into 2 chapters of my dissertation. So because we didn't have any baseline information in Pensacola Bay, it's important to kind of quickly highlight what was known about other areas for comparison. So here where I am now in Sarasota Bay, we've been tracking animals in this area for 50 years. It's the longest longest running bottlenose dolphin program in the world. And over 50 years of data, we know that animals live here all year round. For the most part, they don't really go anywhere. There's a little bit of seasonal variation in terms of where they spend their time, but there's no seasonal influx of animals in this region. Occasionally, individuals will get up and move to other areas like Tampa Bay, but generally we have a stable resident community here. In very stark contrast, a colleague, Brian Balmer, did his work up in St. Joe Bay and showed really strong seasonal movement patterns into St. Joe Bay, in addition to a resident community that lived there all year round. So it's very different than sort of the golden standard of what we know down here in Sarasota Bay. And then again, a slight difference, Rennie Tyson did her master's over in Apalachicola Bay and showed that although there was some evidence of residency here in Apalachicola, over in St. George Sound, most of the animals over there were transient, so they were only ever seen once. In fact, over almost 50% of her animals were only ever seen once. And there was very little movement between these two regions, and only 3.5% of the dolphins that she saw over a 4-year period were ever seen in both areas. And this is over a geographic scale of only about 80 to 100 kilometers, so it's very small scale. And once again, slightly different, my colleague Steve Shippey, who was doing his PhD up here in Choctawhatchee Bay, had slowly started extending his surveys into the lower parts of Pensacola Bay system. And given the work that he was able to do, he showed quite a bit of movement back and forth between these two pretty large systems. So the question mark is, what else is going on with the rest of Pensacola Bay? And let's get in there. So that's where I came in. So we had some very basic population dynamic questions. Are there seasonal changes in abundance, the way Brian Balmer showed in St. Joe Bay? Is there evidence of reduced survival rates in post-oil spill years? And is there evidence of resident community? We started mark recapture work in 2013. Mark recapture, if you're not familiar, is going out and quote unquote capturing an animal once and then recapturing it over time. For us, capturing a dolphin isn't literal, it's just taking photographs because we can track individuals over time using the unique patterns of their dorsal fins. So you can see here in this image, this is the same animal over a 6-year time period. There's very little change in the nicks and notches on the trailing edge. There's a little bit of a tip nick, carving out at the top here, but otherwise we can track individuals like this over time. And so we go out on our boats and conduct surveys throughout the Pensacola Bay system over a 3-day time period, taking photographs and then doing lots of time in the office matching them over time. And we can look at a variety of population dynamics with this approach. We did this for 3 years, and you can see at the bottom here some spaces in the seasons that we were able to cover, but every season was covered twice was the goal. I'm not going to get into mark recapture modeling because that's really complicated, but basically we do a lot of math to try and figure out what the best estimates of abundance and survival are. And because bottlenose dolphins are really complicated socially, if you want to provide an abundance estimate for management, you need to consider whether or not you are capturing animals that really live there full time, or if you have a bunch of animals that came in and are never seen again, that's going to completely blow your estimates out of the water in terms of accuracy. And if you have a dolphin that comes in once and is never seen again, from a modeling perspective, it looks like it died, when in biological relevance, it actually probably just left the system, and that wasn't its home range, if you will. So we used an age-dependent time since marking model, which is relatively underutilized in our work. Actually ended up being the best fit for our purposes. And what it does is it separates modeling components for animals that were seen once ever compared to dolphins that were seen multiple times, and allows us to hone in a little bit on survival estimates by separating those two groups. Overall take home is that abundance in our area did not vary much seasonally, so different from what Brian Balmer was showing in St. Joe Bay, but more consistent to down here in Sarasota Bay. We had quite a bit of error associated with these though, so there's a bit of work to do to try and reduce error. Part of it has to do with the low capture probabilities, for those of you who might be familiar with mark recapture. And our survival rates, basically what you're going to do is ignore this 0.61, because those are the dolphins that we only ever saw once, and that's not going to be accurate. But it allows us to slightly improve our accuracy with these that we saw more than once. Our survival rates were estimated about 0.97 for the animals that we presume were more important to our region. And then because we don't have previous data, we really have to compare that to what's known in other areas. So it's definitely on the high end of survival. Barataria Bay down here, which was the hardest hit in the oil spill, had a range of survival estimates from 0.85 to 0.87. And we now know health-wise that they had a really big hit. So if there was evidence of the oil spill or other factors reducing survival in our area, we would expect this to be much lower. And from a modeling perspective, there are potential reasons why that might be artificially high, and we have some work to do to kind of iron that out. But it's a good starting point at least. Looking at residency, did we have animals that live there all year round? That's a big question mark when you're trying to understand how to manage or how to separate these into different groupings. We definitely did have animals that were present all 4 seasons of the year, but only about 12%. We did have dolphins that were resighted in my work that had been present in 2007. So there's definitely longevity to animals that are present in the system, supporting the concept of a resident population. On the other hand, we had a high proportion of transients. So here you're looking at site fidelity, where this is the number of sightings, or this number of animals that had 1 sighting, 3 sightings, 5 sightings, etc. So 30% of our animals were only ever seen once, and the highest number of resights that we ever had was 20, which for a 3-year study period isn't that high. So there was definitely a lot more transients in this area. Because Pensacola has 1 entry point, the mouth of the pass to the bay, if we have a lot of transients in the area, we would expect, okay, well, maybe we just captured a lot of them in the south end of the bay system. So we plotted all the animals that we saw one time to see where they fell out in our system. And we would expect a congregation down here. If we have coastal animals that just pop in, there's a lot of seagrass beds down here, lots of good eating, fishing down here. And perhaps this is a space that's utilized by animals that don't live here. But we did see single-siding dolphins all throughout our system, which is a little bit surprising. And this is something that we want to get into more. But one possible explanation for this is that I mentioned Steve Shippey had done his work in Choctawhatchee Bay looking at the exchange between animals in these 2 areas. And we might just be missing resights because they didn't move offshore, but they moved next door. And perhaps we're not actually capturing the full extent of where our population ranges. And so we're getting a lot of single site, what looks like a transient dolphin, when really maybe our study design should be broader. And the genetic data does support this, which is what we're going to talk about next. So at the time of the beginning of my research, there was really only 2 areas here in red that had looked at population structure at a fine scale. I mentioned down here in Sarasota already. And then Patty Rozelle and her colleagues up in Georgia and Florida had shown fine scale genetic differentiation with a geographic space of only 80-kilometer separation. Since the beginning of my work, several other studies have come out showing additional areas where fine scale structure has been demonstrated. So the Indian River Lagoon over here, South Florida and the Keys, and Biscayne Bay down here, and some unpublished work by our colleagues over in Texas as well. So there's definitely growing evidence that on a very, very, very fine scale, genetic structure is detectable and potentially common. So that was kind of the question for us, is does the system up here match that expectation? And are the current stock designations accurate? Because they are based not on data, but on assumptions, and we wanted to be able to map the data onto that and make sure that they were appropriate. So I was able to capitalize on not only samples that we collected in Pensacola Bay, but with samples available throughout this range from other colleagues, both through biopsy sampling. We use a crossbow and a modified bolt tip in order to collect a sample through biopsies. You can see a plug of skin and blubber down here on this ruler, possibly. It's kind of small where my red cursor is. And that's just the size of your fingertip, really, with blubber and skin that you can get out of a biopsy sample. And then you can also get tissue samples from satellite tagging. So Brian Ballmer's work over in St. Joe Bay, he did some satellite tagging. And when you push the satellite tag in, you can get a plug of skin as well from that method. So over this entire range, we were able to mostly fill in the gaps. And given sort of geographic separation of these areas, had sort of seven clusterings of sample collection sites to work with. One tricky thing about doing genetic structuring, just like population demographics, is this overlap between residents and non-residents in your system. So if you want to look at fine scale structuring and you've got a bunch of animals that come and utilize the area that aren't from there and possibly came from the coast, you could really mask fine scale structuring genetically that way if you include them. So we did a bit of sort of weeding out what we knew about the animals to try and create different data sets based on whether or not we knew something about them. We ended up with 5 different genetic data sets to work with, one being everything that we had, and then starting to remove animals that we knew were non-residents based on other data. So whether it was tracking data or any of our mark recapture work where we attract individuals over time, they'd only ever been seen once, and over a several year period. And the idea was if we could run genetic structuring models across all 5 data sets and they told the same story, that no matter what kind of bias was introduced with all of those different samples, if we had the same story across it, then we would feel a little bit more confident in our results. And that's exactly what happened. There was congruency across all of these. So we're going to be talking about the full data set. For any geneticists or population genetics people listening, we use 19 microsats to work with our genetic data, and worked with the control region for mitochondrial DNA. And we used both types of markers. Microsats tend to give us more contemporary gene flow on a more recent evolutionary time scale, and mitochondrial DNA gives us a deeper time scale on the evolutionary time scale, if you will. So when you use both sets of markers, sometimes they tell the same story and sometimes they tell a different story, which can be really useful. All right, so through a variety of steps to try and understand of those 7 different groupings of areas across the Florida Panhandle, we don't want to tell the models what we think the genetic structuring is going to be. We want the models to take the data and tell us what the genetic structuring looks like. So what you're looking at here is a typical structure plot, the result. And if you're not familiar with what that on Earth that means, these are individuals across this bar here from west to east. So the acronyms at the top are indicating the bay system that the samples came from. So you'll see PCB is Pensacola Bay here. There's a dividing line, and then Santa Rosa Sound dividing line, Choctawhatchee Bay dividing line, et cetera, et cetera. And what we're doing is having the program tell us, given microsatellites, how many distinct genetic clusters we have across that region. Each bar here then is an individual that we sampled. And the proportion of coloring, like if it has lots and lots of red in it, then it's very high certainty that that individual was assigned to the red cluster versus the yellow cluster versus the green cluster. And if you've got different colors on a given bar, then there was less certainty in assignment of that cluster. So through lots of different steps in a hierarchical approach to structure modeling, evidence points to 3 genetic clusters. We've got Pensacola Bay and Choctawhatchee Bay clustering in this red group over here, St. Andrews Bay being genetically distinct from that, and then all 3 systems to the east of that kind of grouping out together, potentially with some mixing here in St. Andrews Sound. And then the goal is to see, okay, is there a significant difference between those 3 groups? and through pairwise comparisons, and yes, pretty strong significant differences regardless of the marker being used. There was no evidence of sex-biased dispersal, which is not surprising given past work in this species. Despite males tending to have larger home ranges compared to females, there hasn't been any evidence of sex-biased dispersal in Tursiops truncatus yet. So at least this is in line with the current research. We also looked at direction of gene flow. And this is a relatively new type of analysis that hasn't been implemented as much in the Gulf of Mexico and Southeast just yet. So we don't have a lot to compare to, but what we would expect given coastal and offshore ecotypes is that in evolutionary timescales, the Gulf of Mexico existed long before the bay sounds and estuaries were formed. So from a dolphin perspective, we would expect founder populations and what we call source populations to be the offshore form. And then over evolutionary time, the bay sounds and estuaries open up and start to form and create habitat available for them to move into. And so on a contemporary time scale, we would expect dolphins inshore to be newer, if you will, on an in evolutionary time. So the direction of gene flow that makes the most sense would be coastal to inshore. What we don't really expect is there to be a lot of gene flow laterally, east to west or north to south, but it's also kind of an open question box because we're only just starting to look at this. So what I'm showing here is these three groups that we defined using structure that I just mentioned, and the arrows are showing direction of gene flow, and the strength of the gene flow is the size of the arrow. So the two in green are showing you that there's gene flow coming into this middle system, St. Andrews Bay, but very little leaving it from an east to west perspective. So we've kind of got a source sink dynamic, or at least evidence for one, within these small bay sounds and estuaries, which is a little bit surprising. And I don't have great explanation for that. We have a lot of work to do to try and figure this out. But I will get into some possibilities here in just a sec. Another interesting part of this was that from using mitochondrial DNA markers, St. Andrews Bay was also genetically less diverse. So these indices are an index of genetic diversity, and you can just see by the numbers that 0.5 is much less than 0.7 or 0.8. If you're a population geneticist, you might be looking at my sample sizes and show that St. Andrews Bay also has at least half less than sample sizes compared to the other two. So we might just not have had enough samples to be able to detect other haplotypes in this system. And one argument against that is even broken down into these individual units that we started with, which is Pensacola Bay, Choctawhatchee Bay, St. Andrews Bay, and so forth, with relatively comparable sample sizes. We still have a really low genetic diversity just for St. Andrews Bay. So what's going on here? One last slide just to kind of show you a general haplotype network. Each of these codes is a unique haplotype that was detected across our samples, and the colors correspond to those three groups that I just mentioned. So in here, we've got TTR2 is that particular haplotype is found in all three systems, versus these two over here, which were unique to the Western Panhandle, and these over here that were unique to the Eastern Panhandle. And we don't have any unique haplotypes in St. Andrews Bay. So one potential explanation, or at least factor, that could be contributing to this is the fact that Pensacola Bay is the closest in distance to deep water in the entire Gulf of Mexico. And the Fort DeSoto Canyon, the mouth of that comes right up to the offshore systems in Pensacola Bay. If you're a fisherman, you know that getting out there, depending on the currents, that blue water, deep blue water can come in quite close to the coast. And that's going to affect marine mammal populations as well, and whether or not those deep water populations are utilizing habitat much closer to the inshore systems compared to, let's say, over here in Sarasota Bay, where I am now, with a shelf that exists for quite a couple hundred miles. A paper that came out as I was finishing this up also looked at Gulf of Mexico genetic structure. So instead of the very nice and neat delineated map that I showed earlier, the expectation was that structuring would follow these bathymetric lines, which it generally kind of does in the east and west sides of the Gulf of Mexico. But right here, where we all are, is a hot mess of overlap between populations, coastal and offshore. And so we might have a situation where some of our inshore systems here in the Northern Gulf are actually being influenced by coastal animals and offshore animals much more than other areas of the Gulf in Mexico, just because of their proximity to each other. And the inshore system having resources that might be more easily accessible to offshore animals. So there's a lot of work to be done here to tease this stuff out. And really what we need is to combine coastal and inshore sampling to really look at this. At the end of the day, part of my project was to inform management units, and given the genetics, we are proposing that Choctawhatchee and Pensacola Bay should be combined into a single management unit. The structure around managing this area here in the Panhandle has been updated already, given Brian Ballmer's previous work. And there is some evidence in my work that this eastern section here over in Apalachicola Bay could still be potentially different. It clustered all together as an eastern grouping with St. Joe Bay, but we also didn't have sample coverage in Apalachicola Bay. And there's a lot more to kind of resolve this in any level of certainty. Okay, looking at time, taking a drink of a glass of water. Give me just a sec. All right, so the last piece of this is a case study that came out of my work. We had a flood in 2014, which, being in Mobile, you guys also experienced because it came right over you guys and dumped on us. And this was 20 inches of water in 30 hours. It was like a historic record-breaking flood for us, lots of damage. And it was pretty impactful, at least visually, for our dolphins in the area as well. So I happened to be out there at the time doing marker capture for my population dynamics work, and we started seeing dolphins that look like this. There wasn't a lot known at the time as to what causes skin lesions, and we didn't have the capabilities of being able to biopsy dart yet. So we weren't able to take advantage of sampling to be able to look at histopathology or anything like that. So we did more of a visual approach to trying to characterize how important this was, in combination with some EPA data that was actually very valuable in helping us understand the extent of the flooding issue. So up here in the upper left is EPA CTD stations throughout Pensacola Bay. Each of these dots is a CTD station that the EPA monitors regularly. And they had put out a paper a while back showing that across these stations, or across the system, there's quite a bit of variability in freshwater flow naturally. So what you're looking at here is a cross section of the bay. And each of these dots in the upper left corresponds to a triangle from left to right across this plot. So this is a salinity depth profile, and you're looking at a cross section of Pensacola Bay and Escambia Bay from the northern section on the left, which is Escambia Bay, to the mouth of Pensacola Bay to the right. So as you would expect a lot of salt water to be coming in from the Gulf, the dark blue is indeed saltier water, which you get this wedge that kind of always exists at the bottom near the pass. And then with different rates of flow coming from the rivers, we get different levels of freshwater lenses that occur naturally outside of the flood. So that was an important thing to know. So dolphins in this area are experiencing potential variability in freshwater exposure, regardless of a flood event like this. Well, we wanted to see what it looked like during the flood. And just for orientation, this black line is just a visual line to show you where Escambia Bay kind of ends, because it's such a shallow system. If there's dolphins in that system during high flow times of year, then they're basically going to be exposed to fresh water top to bottom, as opposed to having some, salinity, higher salinity to get away from it in that particular area. So right before the flood, April 21st of that year, we had already a high flow time period, and actually there was a freshwater event, a rainfall event 2 weeks before the flood. So grounds were already saturated, discharge rates were already high. And then the flood happened, and the CTD station picked it up on May 13th. Flood was on the 28th to 30th or 29th to 30th. So within 2 weeks, there was a freshwater lens blown out all the way to the pass. And according to the folks at EPA that have been doing this a heck of a lot longer than I have described that they've never seen this before, where the freshwater lens extended that far to the pass. And so you can see where the line delineates Escambia Bay. Escambia was completely fresh, top to bottom. And it continued to be that way in June. And in July, we were starting to get some stratification in Escambia Bay, but only up to about 10 parts per thousand. And we now know that general guideline currently is 8 to 10,000 parts per thousand, that dolphins seem to be kind of considered freshwater for dolphins, if you will. So this freshwater lens existed for several months following the spill. It wasn't just following the flood. It wasn't just an isolated event that went away and returned to normal quickly, which we would expect to have potentially devastating effect on bottlenose dolphins because freshwater is known to impact them. And at the time, we didn't really understand much about this, but there was definitely evidence, or it was starting to become known, that dolphins can have suppressed immune function due to prolonged freshwater exposure, and have numerous other potential health concerns that we didn't really understand well at the time. So it could potentially be a real big problem for their health. So again, we didn't have the capacity to do biopsy darting at the time, so we did a visual assessment looking at the prevalence and extent of skin lesions in our system before and after the flood, focusing just on 4 seasons. So the winter prior, the flood, spring, and then 2 seasons after that, covering the 6 months following the flood. And there's a lot of intricacies into how we did this. We actually ended up publishing an entire publication on how to visually assess skin lesions. And so we chose a rating system to quantify extent as a low, moderate, and high, and also used the stranding records that Dauphin Island Sea Lab has and Emerald Coast Wildlife Refuge keeps, so that we could get some idea or a proxy of whether or not there was an increase in mortality rates in response to the flood as well. And there's a lot to be said about what we saw. I'll try and give you the rough take home. Is basically we didn't see the expansive skin lesion outbreak that we expected to see. So of the 333 unique dolphins that we saw at least once over 6 months, only 20% of those were ever seen with skin lesions once over that 6-month period. So again, I'm not talking about my 3 years of data, I'm only talking about 6 months following the flood. And only one of those dolphins was seen with skin lesion extent cover of 50% or more. So all of the rest of the dolphins that we saw with skin lesions, the proportion of the body that was covered with those skin lesions was less than 50%. And of those 333, only 33 were ever seen with skin lesion extent greater than 5%. And we call 5% as kind of our background cutoff because we've discovered that we do have skin lesions on dolphins outside of this flood event in the winter prior. And now we know in other seasons as well. And so there is sort of this background level of skin lesions, but they're so small that we would have never really noticed them because we weren't looking for them at the time. So 5% is a bit of an arbitrary cutoff, but the fact that only 33 dolphins over a 6-month time period, when we had fresh water for several months, were seen with skin lesions was quite surprising. We also had some trouble because many of those dolphins were only ever seen once. So when we're starting to look at like repeated measures and look at changes in skin lesion extent and prevalence over time, that can get tricky. statistically. So the best way to present this visually is to show you that there are 21 dolphins that we saw with skin lesions over time at levels of extent greater than 5%. So these are basically what I would call the most important ones to look at in our system. And this is a visual representation of the changes that these individuals experienced over time. So each code here is an individual ID. This is the rating that they were given during the winter prior to the flood, and these are the codes that they were given after the flood. And what we're seeing is a general trend towards higher extent, which is in red, compared to pale, with no visible lesions were present. Statistically, this was significant as well, but it gets into some weaker evidence just given what we were able to do with our analyses and with sample sizes. We also looked at the stranding data. So we wanted to know, okay, well, maybe they didn't have a big outbreak across the population, but there might be evidence that they still had a pretty big hit to health of the individuals that were impacted. So we split the stranding record across this entire region into 3 groups, which include Alabama, what we call the Florida impacted region, and the non-impacted region. We separated these out based on rainfall. And the reason we separated out Alabama in our section was just because Alabama was exposed to the Deepwater Horizon Oil Spill at much greater impact than we were. And publications had shown that there was already residual impacts from the Deepwater Horizon Oil Spill impacting strandings. We didn't want that to be biasing our study site here. So we looked at whether or not there were significant differences in stranding record or strandings across these 3 regions, and whether or not there was any changes over time, looking at 2012 to 2016, so 2 years before and 2 years after the 2014 flood event. And generally, no, there really wasn't an effect here. The only effect was a regional effect, and the pairwise comparison showed that Alabama was significantly higher in number of strandings than the other 2 regions, which was not surprising given that some of that data was still impacted by the oil spill. So we really didn't have a huge outbreak. There wasn't a large number of dolphins that were impacted. There wasn't a huge stranding event associated with this flood, and yet we still had this freshwater lens for months. And we have other case studies that have shown animals being stuck in Lake Pontchartrain, for example, with extensive skin lesions, and the Stranding Network with you guys, Dolphin Island Sea Lab, has given case studies where animals get these skin lesions and have died with freshwater exposure. So why weren't we seeing that in our system? And we don't have a great answer. But we did start to look at distributional shifts. So maybe, given what the EPA had already told us about freshwater flows and pulses of events naturally in our system, maybe our dolphins are already kind of coped to deal with that. We have shallow northern sections, but we have much deeper waters further south. We have much more like saline influence near the pass that they can get in, out of the freshwater lenses. And we have a big channel basically running that CTD transect all the way up Escambia Bay. It's a pretty deep channel. Perhaps they're able to already cope with this, just having been exposed to it over time. So my colleague Shauna McBride is looking at that currently, whether or not there's distributional shifts in response to salinity and depth. Like, are they moving south towards the pass? Are they moving towards areas of higher salinity to get out of freshwater? And this is still a work in progress, but basically the take-home message here is that looking at starting with hotspots, there really was no shift between, let me back up a second and explain what we're looking at. So this is spring, the year that we got the flood, hotspot analysis, and then spring, not the year of the flood, hotspot analysis as a control for comparison. And if there was any shifts in distribution, we would expect these little red squares to change in location. And then this is mapped onto salinity. And really, between spring and its control, there was no major shifts. We would expect it to be further south or towards like the center of Escambia Bay and Pensacola Bay where the deep channel is. We also did the same comparison for summer months because we had extended freshwater conditions past our spring session, the year of the flood. And again, our hotspot distribution, it really doesn't differ between the impacted summer and the control summer. Similarly, looking at kernel density estimates, we're now looking at fine scale shifts, not just a hotspot, but fine scale shifts from one season to another. So back to spring again, what you're looking at here is spring of the flood compared to its control. And if dolphins were shifting in response to the flood, we would expect them to be moving south or towards deeper areas. So the blue here is indicating from one season to the next, from a control season, they moved out of a blue area, and during the flood, they moved into the red area. So there's a slight southern shift here from blue to red, from control to flood, but this is only a couple kilometers shift. We're not seeing big widespread shifts from like Escambia Bay down to Pensacola Bay, which is what we would have expected. Looking again at summer, we're again looking at during the control season, blue leaving these areas and moving towards the red during the flood season. That's a much stronger shift from these northern areas down to the south, a little bit more like what we would have looked or what we were looking for, but also only in this season so far. She's also doing some species distribution modeling to try and understand which variables are impacting dolphin density. And so far, preliminary evidence is showing that there is higher densities in the south during flood impacted seasons, also with decreased nitrogen concentrations in areas with higher bottom salinities. That's a big mouthful to say that there's some evidence that there's some southern shifts with response to the flood. But this is a work in progress, and we have still yet to complete it. And so she's still kind of ironing out some of this data. The take home is that generally we still haven't been able to explain it. That's the take home. All of the things that we've come up with that we've been able to look at have not really explained why we didn't have more problems or more mortality than we did. Or we just don't have the data to show it, which is another possibility with our data quality. So to wrap up, since I am over time, basically we're finally getting to put everything together across the Gulf of Mexico. We're filling in the gaps. This is what this map looks like now with everybody's involvement over the last decade or so, including our own expansion down here. We're starting to fill in Naples area. And moving forward, we can start to ask some really interesting questions about what really affects demography of these animals and population dynamics. And same with population structure. You know, there's a lot to learn about connectivity and movement and the relationship between these different communities, and also what drives population structure. We really needed to have the data to be able to start answering some of those cool questions, which are coming. And generally speaking, there's still lots to do. As I was finishing up my dissertation, there was yet another unusual mortality event going on in the Northern Gulf last year. And that was mostly attributed to freshwater influx again. You guys have already experienced multiple hurricanes this year. Who knows what's going to happen with that? And you know, the amount of fresh water that's been dumped into Louisiana, Mississippi, Alabama, Florida yet again this year. So this is important stuff to be able to look at and understand. These are a top predator and they're an ecosystem indicator. So while they are not an endangered species, they're really important for understanding the health of the ecosystem in general and connections between ecosystems as well. And what we can learn from them, we can also potentially help to identify problems and concerns in other species where we might not have as good of access to them. So with that, thanks to very, very many people. This is my committee who helped me get through this, and to all the volunteers and students and colleagues that helped me get through my research, including the University of West Florida that let me camp out at their university for 3 years to be able to do this work, even though my home institution was down at UCF, and to my academic families away from my family, which includes Dolphin Island Sea Lab and the Ruth Carmichael Lab. Thank you to all of you for taking me in, and thank you all for being here and listening to me. I appreciate your time. And with that, I will leave you a funny cartoon that makes me giggle when I see it. And I'm done.
That was great. Thank you so much, Christina. We really appreciate you coming and sharing your work with us. For everyone else, I invite you to either feel free to unmute yourself and ask a question, or if you're more comfortable, you're welcome to put a question into the chat.
Ooh, how do I see the chat?
There should be a bar at the bottom. I'm actually looking at the chat right now. Everyone's being very quiet.
Hey, Christina, I have a question for you.
Hi, Matt.
So I was wondering if you happen to see any behavioral changes in the dolphins after the freshwater event, in terms of like, were they foraging more or less, or traveling more, things like that?
We have not gotten that far.
Okay.
So yeah, we haven't done anything with direct behavioral responses with respect to freshwater. That is a question we haven't tackled yet.
Okay, thanks.
Mm-hmm.
Sort of along those same lines, and maybe the answer is the same, but I was wondering if, while you guys didn't notice any distribution changes sort of north-south so much, if there was any way of detecting whether the animals were maybe staying in the same area, but going deeper to the higher salinity water deeper.
Yes, and so that's one of the variables that's included in Shauna's model, especially her species distribution models. So there is evidence that they are shifting towards more saline bottom waters, but our bottom spatial coverage is very minimal. So that is a variable that we just don't have great coverage, and so it's hard to really know how to interpret it. But preliminarily, there is evidence, given her models, that they are using the deeper areas a little bit more during those flood impacted seasons.
Hi, Christina. This is Lauren Clance. We haven't met yet, but I'm also in the Carmichael lab and doing dolphin research in Mobile Bay. I was curious to know, for the dolphins that you did see those lesions on, was there a dominant type after the flood?
So that comes back to how we categorize them, which is a whole talk in itself. So yes, sort of, kind of, depending on how you categorize them. So what we got away from a little bit was trying to give them specific names and categories, because as we were doing this, we realized that we didn't really know how to best categorize them, because we didn't have the ability to do any pathology on them. And we were doing a lot of hand waving to say what things were. So we did very general categories, and the highest prevalence, if you will, would have been those what we called potentially infectious versions of lesions that we know were either pox virus, or we had veterinarians and other pathologists tell us were potentially directly freshwater lesions given their their experience. So we kind of had this broad category of potentially infectious, herpes virus, things like ring lesions, if you will, or what are we calling them? Fringe lesions. So that's been pretty common term in the literature is black fringe or white fringe or pox virus spots. Those were the most common collectively. And then we kind of categorize them into all other types of hypopigmentation, all other types of hyperpigmentation, and then sort of some other categories, including rank mark associated and stuff. So my methodology publication, which I'd be happy to send you, talks about that a bit more. And we just submitted this research report for publication, but in my dissertation, it breaks it down a little bit more, which I'm also happy to share if you want to take a look at that.
Great, thanks.
Yeah, that would be fantastic, Christina, if you don't mind sharing that, because we're trying to do something similar with animals from this past year and looking at freshwater from 2019 and 2020.
No problem. And Ruth, I think you have my dissertation, but I have probably an updated version of that chapter with the report, so I'd be happy to send that to you guys.
Do we have other questions? Well, I have one more. When you were doing your abundance estimates, did you do any detection bias corrections associated with, because you have so many animals that were only seen one time, and I was wondering if that was something that you considered in your calculations?
Did we do any detection bias calculations? You're making me think about what we did. So for abundance, we did not, well, let's see. So we did some unmarked proportion calculations and adjustments. I'm gonna have to get back to you on that. I can't remember. It's been too long. I don't remember us adjusting abundance estimates for our transient group, but I would have to go look because I do not remember.
Okay, thank you. All right, we do have time for a couple more questions.
I'll go again. Do you think there's a lot of overlap between the dolphins in Pensacola Bay with the ones in Perdido Bay and maybe into Mobile Bay?
Yes, that's my presumption. So I would expect Perdido Bay especially, and I would love to see what kind of overlap you guys get with us at some point. I think just based on ecology and general geographic features, I would not be surprised if Mobile Bay, given its size and very different, I don't know, ecological structure with all your freshwater and everything else, I wouldn't be surprised if you guys had your own population that would probably maybe even be genetically distinct, given that the pass to Mobile Bay is not that far away compared to things to my east. And I can't remember if Perdido Bay is an artificial connection historically or not, but especially if that's an artificial connection that was only dredged out for the ICW, then Mobile Bay, just hypothesizing forward, I would expect to have a unique population, but definitely also expect there to be some movement in between there. And the thing that was interesting one of the reasons I think Pensacola and Choctawhatchee are more connected is because Choctawhatchee Bay historically did not have a natural pass. So it was a closed system until we opened it artificially. But the Santa Rosa Sound Lagoon connecting the two was natural. So the only way for a dolphin to get to Choctawhatchee Bay historically was to come into Pensacola Bay or somewhere else from the west and come into Choctawhatchee Bay from that direction until we opened that pass up. And so I think that creates more of a natural movement of animals into Choctawhatchee Bay as it was becoming more habitable. And it was originally more of a freshwater system as well until we created more of a marine system with that opening of the pass. So that explains why there's a lot potentially of movement between those two systems. And it would be really interesting to see what would, how much connectivity there would be to the west as well.
Awesome, thank you.
Well, thank you so much, Christina. We really appreciate your time. And maybe if you don't mind hanging on for just a couple minutes, we can say goodbye. And if anybody has any lingering questions and they maybe just want to ask you privately, we can just hang out for a minute or two.
Sure.
Great. Thank you, everyone. We'll see you next Friday.
Thank you all for your attention. I appreciate it.